Good morning, everyone. Thank you for joining to the presentation. My name is Dinesh Wellawa. I'm a graduate student affiliated to Vaccine and Infectious Disease Organization and to the Department of Veterinary Microbiology in University of Saskatchewan. My topic is tackling Salmonella epidemics in poultry using Angel's Glow. So what is Angel Glow? We'll see in shortly. So particular Salmonella seroa that we are working or we are interested in is called Salmonella enteritidis. It is the #1 foodborne pathogen that causes salmonellosis in Canada. It's a gram-negative bacteria. Here is a scanning electron microscopic image of the Salmonella enteritidis strain that we have. So it's a rod-shaped bacterium, as you can see, and those tiny protrusions coming from the surface are called fimbria that help them to attach to cell surface. And this whip-like structure is called as flagellar apparatus that help them to move. So Salmonella enteritidis is considered as a successful intracellular pathogen, so it can invade the epithelial cells and they can persist in the epithelial cells, and also it can invade immune cells and cause systemic infection. So, but the main clinical manifestation of Salmonella infection is a gastroenteritis, and it clinically apparent as diarrhea. But certain percentage of Salmonella enteritis, like 5% of cases, can go systemic infection. Once it goes systemic infection, then can have fatal diseases ranging from meningitis to pneumonia. So what is Angel's Glow? Angel's Glow is known as a bacterial, refers to as a bacterial bioluminescence. During American Civil War, some of the soldiers came up with wounds that glow in the dark, and later they found out there was a particular bacteria called Fort Habdus luminescens, which create a light, was contaminated in these wounds. And they found out that the wounds that was contaminated by the Fort Habdus luminescens heal so faster than the other non-glowing wounds. That's because of these Fort Habdus species can secrete some of the toxin that can kill other bacteria around the wound. So it's accelerated the fast healing. So it's called as Angel's Glow. So the Fort Habdus luminescens has a symbiotic relationship with this nematode that lives in land. So how does the photohabdus creates a light signal? So they have genetic information that can catalyze the chemical reactions inside the cytoplasm of the bacterial cells. So this is encoded by sort of 5 set of genes on naloxone. So I have exemplified the chemical reaction as this. So the reduced flaming mononucleotide can be oxidized in the presence of luciferase enzyme, and that can create a blue to green light, emitted light. So if you have a photo detector that can detect this light signal, you can even track the pathogens and also even quantify the amount of signal that it produces. So our objective of this particular study is to use that information of the genes that are responsible to make the bacteria to glow in Fort Habdus and try to incorporate into a dark bacterium like Salmonella. Theoretically, if you can incorporate under a suitable promoter or express this set of genes under a suitable promoter, you can generate a light signals. So that's what we did. We created a bioluminescent Salmonella strain using that information on the photohabdus luminescence. So the information is encoded in lux C, D, A, B operons, and the lux A and B encoded for that luciferase enzymes, and the rest of the genes enable supply the substrate for the chemical reaction. So it's recycled inside the cells. So utilizing a mini TN7 based system that's adopted and developed in vitro, we can incorporate this set of genes into specific Salmonella chromosome called ATT TN7 sites. Once you incorporate into the bacterial chromosome, it will not affect the downstream or upstream gene of into the won't affect the upstream or downstream genes. That's why we utilize this mini TN7 system in order to incorporate these kind of genes. So the reference of this protocol I'll be presenting at the end of the presentations. So our overall objective of this project is to utilize those bioluminescent Salmonella strain to develop an imaging model using chicken as a model. So why we want to track pathogens once inside the host? If you think about it, if you can generate a model that you can track the pathogen in a live bird without killing them, you can actually study the kinetics of the disease progression using a single bird or couple of birds, rather using 100 or 50 birds in a particular experiment. The same model can be utilized to accelerate a drug discovery, to find out the response to a particular drug, maybe antibiotics, or some of the vaccines. So it's a very fast and convenient model and very cost-effective model. For example, in this case, I have given an example that's how the cancer researchers utilize this system. So once implanted this breast cancer tissue under this specific mice into subcutaneous tissue, and then you can they monitor how this cancer cell progress into a metastatic form. You can see after the week 9, once it was metastatic to the other regions like this axial lymph node in the armpits. So it's a very good information that gives out what is the rate of the metastasis and where it also goes, and so that helps us to streamline the kinetic of the cancer cells progression, and also develop different vaccine by looking at just a couple of mice. So same techniques, that's we are hoping to generate the same technique or utilize the same technique in using chicken as a model. So we challenge the chicken with the 10 to the 9 dose of bioluminescent salmonella strain, and we euthanize each bird for a different time point and try to image whole animal, and also we try to do an ex vivo imaging as well. Unfortunately, we could not get a good signal from whole intact birds because the light signal created is either weak or it's most and also the anatomical barriers that the light has to pass through in the bird, because they are relatively large size rather than mice. It's hard to detect a good signal from the surface from intact bird. But when you take the ex vivo or take out the gastrointestinal tract, and we can see even early as 2 hours infection, you can see some signals coming from, and 4 hours in the small intestine, again in 8 hours very rapidly it's go to cecum, and the same signal will coming from again when 30 hours for another bird, and resembling that cecum as a primary colonization site. Apart from the cecum, you can also detect signals coming from the crop regions, then and indicating that's another apart from the cecum, the crop can be act as a replication niche. So the detection limit of this particular experiment was around 10 to the 6. So you need to have around 10 to the 6 CFU gram of tissue of bacterial in order to get a detectable signal in these compartments. So another experiments we did at parallel, and one is that utilizing different virulent Salmonella introduced strains. We want to observe what's the kinetic of the colonization pattern. And also we created some of knockout genes of interest of this particular virulent strain to see if we can detect any colonization defect compared to the wild type strain. So here in the top left-hand corner is the wild type strain tagged with our reporter system, and we infected the chickens and we look at the colonization pattern. You can see it can cause yolk sac infections after day 4 post challenge, and also can colonize in the cecum and the colon region, and some signal from the ileum regions. And compared to the wild type, we tested different mutant of the same strain and the same constitutive promoter utilizing the same promoter. You can see that if I ask the audience, try to find the colonization differences between the compared to the wild type, I'm sure that most of them you can jot down several differences within under 1 minute time, without any prior knowledge what sort of genes I have been knocked out in these each images that I have displayed here. So in this case, we found out that these 2 genes that is knockout, the delta stand for the knockout of the gene, the SPY1 that stand for Salmonella island 1, and the first stand for the ferric uptake regulator. Specifically, we count the colony count, we found out when you knock out the ferric uptake regulator, which stand for the regulation of the iron uptake in Salmonella, that significantly affected the colonization of the in the yolk sac. So that gives a researcher indicating that's a target, or the iron uptake system can be a target for faster drug discovery, and also good antigens for utilized in developing vaccines. So in the conventional model, we confirm this information. So 75-80% of the birds were not infected in the yolk at all because of this mutation. So it's a very powerful tool. So in the future, we have to improve the detection limit of the current reporter system, because I already mentioned 10 to the 6, and hopefully if we can go to 10 to the 5, that's a good improvement. And also we are currently using it as a in vivo gene expression studies, and also we are using is to mark the different salmonella strain. You can eventually do competitive studies, which is a classical microbiology study to find any fitness defect or compare the virulence of the different strains. And also if you can if you count colonies in a plate, you can do imaging on the plate, and you can count you can detect the colonies like this as a heat map shown in here. So you can count the colonies, and you mark the strain of interest with the bioluminescent strain, and you can image a plate and count and can differentiate from other strains. In this particular image A stand for number particular experiments. We try to find out the cecal colony count from particular birds, and for particular experiments it used the commercial broilers. In commercial broilers, we use the cecal counts and do dilutions and plated them in the brilliant green plates. Brilliant green agar plates gives pink colonies for salmon if they are salmonella colonies. But in this particular experiments, once we challenge, we cannot even hard to get a dilution count because of other E. coli strains hampered our colony count. They were overgrown, and the plate color was changed. And in this particular plate in number 25 is the bird number, the dilution 2. It's normally this is dilution we have to be counted, but we cannot even count because there's a lot of other E. coli strains as obscuring the detection of salmonella colonies. So when we do the image in here, this image results, you can even count easily 41 colony count. So in this particular, so it's a practical issues. We alleviate using bioluminescent imaging. You can alleviate those kind of practical issues when doing colony counts. So with this, I want to wind up my presentations. I especially thank my supervisor, Dr. Wolfgang Koester and Shirley Lamb, for their guidance, and my committee members, Dr. Aaron White, Heather Wilson, and Brenda Allen. And Dr. Aaron White and his team at VIDO developed the initial plasmid and vectors to incorporate this reporter system into another salmonella strain called Salmonella typhimurium. So we utilize the same system what they have developed. So we are thankful to our funding organization, Chicken Farmers Saskatchewan and Canadian Poultry Research Council, for stepping up with the research and giving us the financial support, and also DeVos scholarship to help me with my stipend. And the video is funded by Influence Saskatchewan, and also Minister of Agriculture in Government of Saskatchewan, and also innovation.ca. And this is the important reference that if anybody wants to incorporate a reporter system into gram-negative bacterium, you can utilize this reference. And if you need plasmid and the construct, we are happy to provide it. Thank you for listening, and happy to answer any questions you have.
All right. Well, thank you very much, Dinesh, for your presentation. And thank you for joining us on camera. I know it's difficult when you're all masked up there. Thank you for that. All right, we have quite a few questions. So the first question is, how long does the luminescent signal last?
Thank you for the questions, and thank you, Classing Committee. The signal actually lasts for unless the bacteria die. I mean, it's a constitute express. So you can so rather than the non-conventional method, this can once they are very live, and the bacteria are live, you can detect signal. Once the bacteria is dead, then of course the signal is gone.
All right. And next question, I may have missed it, but what was the detection limit in the tissue for the labeled stain?
That's a good question. The detection limit can be varies with different age of the birds and also the size of the birds. So for the moment, what we have standardized is young birds. We need around 10 to the 6 CFU gram of bacteria in a tissue to detect the signal. We cannot detect signal in liver and spleen when they go systemic. It's because the count is really low. And other factor that's affecting the count value is the materials inside the cecal. Some of the materials inside that's also affect. Sometimes in an empty cecum, it will be appear maybe the detection limit can be low, maybe 10 to the 5 or something like that.
Yeah, you mentioned in your talk that was something that you wanted to work on. So we
have actually improved to 10 to the 5. So those are actually, we are preparing manuscripts. So those data is not available in this presentation.
Oh, perfect. Okay. Also, why do you think that toner gene is relevant to colonization?
I didn't get that. Sorry.
The question is also, why do you think the TONR gene is relevant to colonization?
The TON gene or the FUR?
TON.
Oh, TON-B, okay, TON-B stand for this, if you knock out the TON-B, actually, basically, the ferric uptake is hampered, basically. So the TON-B, what we detect is that even still you knock out the TON-B, it's still colonized. That's very interesting finding. We found out that maybe they have other iron uptake system. They can like especially iron 2 probably abundant, or they can utilize other iron uptake systems apart from the ferric to survive.
All right, good. Are feathers a problem in detecting the luminescence in birds?
I didn't get the questions. Can you repeat it?
Are feathers a problem to detect luminescence in birds, in live birds?
Yeah, I think the most difficult part is the other anatomical barriers like the skin, the breast muscles, those muscles tissues. They actually sort of absorb or just hinders the signal detecting through. But the skin, I don't think the feathers that might be a problem, and there's no autoluminescence from feathers or any other tissues that we detected. There's no, so that's a one advantage to have a bioluminescence rather than the fluorescence.
All right.